We've now talked about how we can use glucose, or fructose or galactose, to produce energy, either 30 ATP per hexose in aerobic conditions, or 2 ATP per hexose in anaerobic conditions. But what happens if we have enough ATP already? We certainly don't want to keep churning through glycolysis and using up all of our glucose. We want to store it for later. In case you forgot it from chapter 18, the major way that humans store energy as carbohydrates is the polysaccharide glycogen. So we need to be able to make that from glucose, and we need to be able to harvest the glucose later. When we make glycogen, the process is called glycogenesis. Notice that this name includes glycogen, which should be a giveaway that it involves glycogen. The name also includes the word genesis, which I hope many of us know means beginning. Many anabolic pathways that are building new large molecules from small molecules will include the word genesis. When we harvest glycogen, the pathway is called glycogenolysis. Again, the word glycogen is part of the name, which should suggest to you that it involves glycogen. Lysis is also part of the name, which is common for catabolic pathways. For example, glycolysis. We'll talk about each of these pathways in more detail, starting with glycogenesis, the anabolic pathway that builds glycogen. Glycogenesis always occurs when we have more glucose than we need for energy. Cells will still pick up the glucose and phosphorylate it in the same way they would for glycolysis, so we can start from glucose 6-phosphate. We've already spent 1 ATP, but it's one that we would have spent for glycolysis anyway. The phosphate needs to be moved to make the hemiacetal more active for glycogenesis. The glucose 1-phosphate formed in the last step needs to be further activated, and to do that we use a uridine triphosphate, or a UTP. Because the UTP loses 2 phosphate pieces at once, this is the equivalent of losing 2 ATP. But now we have a much more active UDP glucose complex. The UDP activated glucose can bind with an existing glycogen chain to increase its length by 1. It costs 2 ATP per glucose to do this. Notice that this means that if glycogen is being consumed anaerobically, we get a net yield of 0 ATP. This doesn't matter because we're always synthesizing the glycogen when we have excess energy. The 2 ATP cost is negligible when we have plenty of energy. And the 2 ATP gain in anaerobic conditions is when we have no other options. Once we have glycogen, we can use it whenever our glucose levels drop low enough to necessitate releasing more. Glycogenolysis is the process by which we break down glycogen to make glucose when glucose levels are low. We're never going to have both glycogenesis and glycogenolysis active at the same time. They're reverse processes. We actually gain a phosphate on the glucose when we break off the glucose from glycogen, which is the equivalent of gaining an ATP. If the glucose is going to be used for glycolysis in the cell, that phosphate just needs to be moved back onto the 6th position, and then the glycolysis can continue. If the glucose is going to be used elsewhere, the phosphate is removed without energy gain, essentially losing an ATP. Again, we get slightly less energy back from the glycogen than we put in to make it, so glycogenesis and glycogenolysis will never be occurring at the same time. Again, it's important to see these pathways together so that we can see how they relate. Every pathway we've talked about, glycolysis, glycogenesis, and glycogenolysis, all start the same way. Glucose gets locked in the cell by phosphorylation. If there's lots of energy already, the glucose 6-phosphate will be converted to glycogen. If there isn't as much energy, the glucose 6-phosphate may be converted to energy in glycolysis. If glucose starts to drop low enough, glucose 6-phosphate can be made from glycogen in glycogenolysis and then either be used in glycolysis or exported to other tissues. We've talked about how we can break down glucose for energy and how glucose is able to produce energy anaerobically by fermenting pyruvate. In humans, pyruvate fermentation yields lactate, or lactic acid, the conjugate acid. The problem is, this lactate can't do anything for us in an anaerobic environment, and it just builds up, which in turn drops blood pH. Our bodies solve this by taking lactate from the anaerobic tissues, which are most likely muscles, and moving them into our liver. The liver tends not to go anaerobically as quickly as the muscles and tends to use fats as an energy supply. The liver will convert lactate back to pyruvate and then use the energy from fats to build pyruvates back into glucose. That glucose can be returned to the muscle tissues and anaerobically digested. The whole process, the Cori cycle, prevents lactate or lactic acid from accumulating to the extent that they otherwise might and allows us to keep a steady supply of glucose while we're exercising. But this process requires a reverse of glycolysis, where pyruvate can be converted back into glucose. That process is called gluconeogenesis. Gluco for glucose, neo for new, and genesis for beginning. Glucose new beginning, gluconeogenesis. Again, notice that the anabolic pathway here has genesis as part of the name. Gluconeogenesis is in many ways the reverse of glycolysis, but not exactly. The jump from phosphoenol pyruvate to pyruvate is so favorable that we can't go directly back the same way. We need to create oxaloacetate first, and then phosphoenol pyruvate. This costs way more energy than we get from glucose. Notice it costs 6 triphosphates, 4 ATP, and 2 GTP, while we only get 2 ATP going the other way. We'll always be going through gluconeogenesis in a different tissue from glycolysis, and it will always be powered by catabolism of a different type of molecule, typically fats. If we don't have these separated, we'd run into a problem we call a futile cycle. We keep on going from glucose to pyruvate and then back, but we'd lose energy every time. We've now talked about 4 pathways relating to carbohydrate metabolism. They all start with G and are all very similar. I'm sorry, I didn't name them. It's still really important to keep these straight. 3 things that may be helpful. 1. Every catabolic pathway has lysis as part of its name. Remember that these are the pathways that go from large to small. Every anabolic pathway has genesis as part of its name. Remember that anabolic is small to large. And 3. Every pathway that involves glycogen has glycogen as part of its name. This can help at least a little bit. In addition to these 4 pathways, note that we also have several possible routes for pyruvate. Anaerobic to squeeze out a little bit of energy from glucose if we don't have oxygen, or aerobic to get way more energy from the citric acid cycle and the electron transport chain. There is a 5th pathway relating to glucose, the pentose phosphate pathway. The pentose phosphate pathway is a catabolic pathway, similar to glycolysis. However, glycolysis is being used to supply energy either anaerobically or aerobically in the form of ATP. The pentose phosphate pathway specifically gives us energy in the form of a reduced cofactor NADPH. The structure of NADPH is shown on the left. The only physical difference between NADH and NADPH is that NADPH has a phosphate on the 2' position of the ribose. This allows different enzymes to recognize and use NADPH than NADH. NADPH is used specifically for synthesizing fatty acids. We'll talk more about fatty acid synthesis, also called lipogenesis, genesis for anabolic pathways, in the next chapter. This figure shows a visual summary of what we've talked about in this chapter. When you ingest carbohydrates, they are digested to monosaccharides, mostly glucose. Glucose can be used for energy or stored for later. When glucose is used for energy, it typically goes through glycolysis to pyruvate, which can be metabolized aerobically in our central metabolism or anaerobically for far less energy. It can also be catabolized through the pentose phosphate pathway to get NADPH specifically to power fatty acid production. If those 2 pathways are active in a cell, glycolysis and the pentose phosphate pathway, we probably have more than enough glucose. We will probably also try to store some for later as glycogen through glycogenesis. On the other hand, if glucose is low, we may break down glycogen through glycogenolysis, and in the liver, we may synthesize glucose from pyruvate with gluconeogenesis. There's quite a bit of logic behind when these pathways are working. When we need energy, the pathways that give us energy are likely to be active. When we need glucose, the pathways that give us glucose are likely to be active. when we need to get rid of glucose, the pathways that use glucose are likely to be active. All of this lays an important foundation for understanding hormonal control of metabolism. We have three major hormones that regulate our metabolism in really broad ways: insulin, glucagon, and epinephrine, which is essentially the same as adrenaline. Insulin is the hormone that says there's too much glucose, so it activates everything that uses up glucose: glycolysis and the pentose phosphate pathway as catabolic pathways, but also glycogenesis, making glycogen, as an anabolic pathway. It will cause everything that makes glucose, like glycogenolysis and gluconeogenesis, to stop. Glucagon is the opposite of insulin. Instead of triggering the use of glucose because there's too much, glucagon signals glucose is gone and triggers the retention and synthesis of glucose because there's too little. It will slow down glycolysis, prevent glycogen synthesis by glycogenesis, and prevent the pentose phosphate pathway. It will also activate glycogenolysis. It also activates gluconeogenesis to make more glucose if muscle tissue is anaerobic. Epinephrine, or adrenaline, is a little bit different than those. Epinephrine is a way of triggering a sudden burst of energy. Think an EpiPen. Only catabolic pathways are activated, and pretty much all the catabolic pathways are activated. Everything with a lysis. So glycogenolysis is going to give us glucose for glycolysis, and we'll also see later that epinephrine will activate catabolic pathways for fats too. Again, anything to give us that burst of energy for a fight or flight response.